Today I would like to start the lesson where we have stopped last time. In the last class we discussed about food components. Our first lesson name is food components. In the last class we discussed about how types of components and their importance. We all know that about the how many types of components. 5 types of components. Components are divided into 5 types. They are carbohydrates, proteins, fats, vitamins and minerals. We all know that about components and their importance. Now coming to the test here. Already we discussed that carbohydrates are the rich source of energy and they are present in potato, wheat, rice, sugar. How can we identify that these carbohydrates are present in potatoes, rice, wheat, sugar? How can we came to know that these are present in that food substances only? By doing simple experiments we can observe the carbohydrates are present in that food sources only. Okay, now coming to the test. Today we are going to discuss about test for carbohydrates, test for proteins, test for fats. Now coming to the first test, that is test for carbohydrates. Test for carbohydrates. Before going to the test for carbohydrates, we must follow 4 steps. What are they? They are first aim, apparatus, procedure, result. Aim. What is aim? To observe the carbohydrates present in the given sample. Whatever the sample we have taken, in that sample we are going to, what is our aim? We are going to observe the carbohydrates whether present in that food source or not. That only our aim. So that only here aim means what you want to write, what you will write, to observe the carbohydrates present in the given sample. Next, apparatus. Here apparatus means materials which are required to observe the carbohydrates. Materials or equipments which are required for the test for carbohydrates. Here mainly test tubes. We are taking the main food source here we are taking as potato. Next one is iodine solution. One of the chemical substance, iodine solution, we are taking as a indicator. Here iodine solution is a indicator. What is an indicator? An indicator is a chemical compound that changes the color and the structure of the substance. Here with the help of the iodine solution only, we can observe the carbohydrates present in the given sample. With this chemical substance only, we can observe carbohydrates are present in the given sample. Next, procedure. First of all, follow the procedure. Take one test tube. Into the test tube, add few drops of iodine solution. We must the iodine solution is more concentrated, so that only we should dilute the iodine solution. How can we dilute it? By adding some water, little quantity of water. By adding little quantity of water to the iodine solution, then the iodine solution is diluted. Keep the dilute iodine solution aside and then take another new test tube. Clean a new test tube. Take the raw potato piece, small piece of raw potato or half piece of raw potato, whatever it is your wish. Take the potato piece into the test tube. Then add the dilute iodine solution into the test tube. Then what the changes color? Here look at this picture. What the color you are going to observe? Yes, isn't it? Dark blue and black color indicates the presence of carbohydrates. That means the potato changes to dark blue and black. Dark blue and black, it indicates the presence of carbohydrates in the given sample. Ultimately, and by that means by observing if we observe the color of the potato, the potato color changes to dark blue and black, so that only we can say that the carbohydrates present in that. What is the ultimate result? By adding the iodine solution, I observe that. What is the result? I observe the color of the potato changes to dark blue and black. For that I came to know that the carbohydrates present in the given sample potato.
Start.
Next test for proteins. One of the important test. Next, test for proteins. How can we identify the given samples? For the test for proteins also, we should follow the four steps. First one, aim. What is our aim? To observe the proteins present in the given sample. Next, apparatus. Here, apparatus means test tube, again test tube holders, but here we are taking the indicators as copper sulfate solution and sodium hydroxide solution. Next, again procedure, and next again result. Coming to the apparatus, we are using chemical substances as copper sulfate solution and sodium hydroxide solution. We must prepare copper sulfate solution 2% copper sulfate solution, and sodium hydroxide solution 10% of sodium hydroxide solution. Copper sulfate 2% of copper sulfate solution we should prepare before going to do the experiment. Then how can we prepare copper sulfate 2% of copper sulfate solution and 10% of sodium hydroxide solution means, first of all, take 2 grams of copper sulfate solution, 2 grams of copper sulfate into the test tube and add 100 ml of water. 100 ml of water. That means to prepare 2% of copper sulfate solution, we are taking the test tubes and to the test tube add 2 grams of copper sulfate solution and then add 100 ml of water. Then it will become 2% of copper sulfate solution. Next again, sodium hydroxide solution. How can we prepare 10% of sodium hydroxide solution means, take 10 grams of sodium hydroxide, 10 grams of sodium hydroxide into the test tube. To that again add 100 ml of water, 100 ml of water. That means you must take 10 grams of sodium hydroxide chemical substance into the test tube. To that add 100 ml of water. Then it will become 10% of sodium hydroxide solution. Here 2% of copper sulfate solution. With these two chemical substances, we are going to do the experiment. Next, in the test for proteins also, you must take the test tubes. To the test tube, we add some milk. Milk are the rich source of proteins. Here we are taking as a milk. To the milk add 2 drops of copper sulfate solution. Just 2 drops of copper sulfate solution and 10 drops of sodium hydroxide solution. Already we prepared 10 sodium hydroxide solution and copper sulfate solution. In that copper sulfate solution, we are adding 2 drops of copper sulfate solution and 10 drops of sodium hydroxide solution to the milk. Then observe the color. If the color changes purple or violet, it indicates the presence of proteins in the given sample. That means by the result, what will be the result? In the milk, the presence of proteins by the color observing, we came to know that the proteins are present in the milk. Because the milk changes into violet or purple, it indicates the presence of proteins in the given sample. Test for fats. This test we can do ourselves. It is very simple test. We can do it in our home. Whatever means, take one piece of paper, empty paper, white paper, and drop 2 or 3 drops of add it to that white paper. Pour 2 or 3 drops of oil to the paper. If the paper changes to translucent, then it indicates the presence of fats in the given sample. You are going to observe that, isn't it? Yes. What you are observing? Just yes. That means if the changes, that means we can see everything by this. That means we can see the transparency is seeing here. So that only if the paper changes to translucent, it indicates the presence of fats in the given sample. See, test for fats. In this test for fats, I poured little quantity of oil in the middle of the paper. Did you observe that? The remaining part where I didn't pour oil, the remaining part is not visualized. only the middle part where I poured the oil and rubbed it gently, that part only visualize it. That means we can see the under paper book, we can see clearly, isn't it? Under the parts present in this paper we can see clearly, but where the oil is dropped and poured in that area only we are going to see. That means if the paper changes to translucent, it indicates the presence of fats in the given sample. Next, these are the three we have discussed up to now. First one is test for carbohydrates. Carbohydrates, what are the how can we say that carbohydrates are present in the rice, wheat or sugar? Means if the substance changes into dark blue or black, it indicates the presence of carbohydrates in the given sample. Next, how can you say that proteins are present in the milk, egg or whatever the rich source of proteins? Means how can you say that? By adding copper sulfate, 2 drops of copper sulfate solution and 10 drops of sodium hydroxide solution, the milk changes into violet color. It indicates the presence of proteins in the given sample. Next, test for fats. How can you say that the fats are present in the given sample? By adding or by pouring little quantity of oil to the paper, white paper, the paper changes to translucent. That means under the paper objects are we can see clearly, but where the oil is not poured in, that area is not visualized on the only the area where we poured. That means where we dropped oil droplets only, there only we can see the paper changes into translucent. These are the 3 experiments we can study up to now. In the next class we discuss about remaining